Welcome everybody to the Gulf Coast Consortia Regenerative Medicine Conference. We've got a busy day ahead of us with a lot of outstanding speakers. We're going to be using the Brella platform to live stream the conference and also use it as a chat and other networking platform. And I want to turn it over to Dawn, who will give you a Cliff Notes version of how to do that and make sure that you're up to speed on that and can take advantage of it throughout the conference. And then after that, I will introduce our first speaker.
Our next rising star is Dr. Renita Horton, and she is a assistant professor in biomedical engineering at the University of Houston. She received her BS in chemical engineering from Mississippi State University and her MS and PhD in engineering sciences with an emphasis in biomedical engineering from Harvard University. Prior to joining UH, she was an assistant professor of agricultural and biomedical engineering at Mississippi State. Her research focuses on understanding the factors that lead to heart disease and sickle cell anemia. She is the principal investigator of the Cardiovascular Tissue Engineering Laboratory that focuses on developing tools and techniques to investigate cardiovascular disease, development, and progression. And today, she's going to tell us about engineering microenvironments for cardiovascular disease models. Dr. Horton.
Thank you so much for the introduction, and good morning, everyone. I've enjoyed the talk so far. Today, I'm going to discuss some of the work that my team has been focused on and with regards to cardiovascular disease models. So my team has a number of different areas, mainly centered around cardiovascular disease modeling. And so one of them is really understanding microenvironments and really how these different microenvironments will change the trajectory of cell fate decisions. So in that case, we're interested in understanding cardiomyogenesis and various microenvironmental factors that drive stem cells towards a cardiac lineage. And the other area that we're interested in is cardiac pathogenesis. And so we're using kind of cues that we're able to define from literature or clinical observations, and then we're building those different factors into our system to then create what we're saying is a in vitro model of disease. And the idea behind that is to improve or provide tools for further mechanistic and therapeutic studies. And the final area that my team looks at is sickle cell disease. And so we look at it from a different standpoint. So thinking about the disease itself, the image that I have here is what's considered to be indicative of or definitive within sickle cell disease, being that you have these red blood cells. Once they become deoxygenated, that leads to hemoglobin polymerization due to a single point mutation within the hemoglobin gene. And because of that, this polymerization causes the red blood cells to undergo a morphological change, leading to the sickling, and hence the name sickle cell anemia. So we're not really interested in really understanding the cascade behind the red blood cell remodeling, but what we are interested in is understanding the consequence of sickle cell disease on the cardiovascular system. So the image that I have to the right shows just a number of different systems that are affected by sickle cell. So this is these are some of the common issues that individuals with sickle cell disease suffer from. And so we're really interested in understanding the cardiovascular side of that. And so we've done some modeling, and then we're subsequently moving into the actual in vitro models that I'll talk about within this talk. So so to try to kind of center the things that we're doing and why we're doing them, really looking at the effects of cardiovascular diseases. And so this is one of the leading causes of death globally. And we suggest that there are not enough tools to adequately assess cardiovascular disease mechanisms. And that's where the area that we're really focused on is really, can we develop tools that can effectively recapitulate features of this disease? And so the ways that we do that are really looking at what's happening within the body, and then picking out specific features that we can effectively model within our system. And one of the things that we can appreciate with regards to the heart is the fact that the heart tissue itself is very well organized. And so the organization of that heart tissue is really important in how the heart functions. So in one case where you see the healthy tissue, you can see that it's fairly well aligned. And so that alignment drives how well the heart is able to effectively pump blood throughout the body. And when an insult occurs, this leads to reorganization within the heart. And so in some cases, that could be a fibrotic tissue that's forming, and that can also lead to ineffective heart function. And so our goal is to really develop platforms to recapitulate disease, to support both mechanistic and reparative studies, and also to look at drug efficacy assays. And so the goal would be to be able to use these platforms as a test bed for potential therapies. So some of the hallmarks of cardiac failure is reactivation of fetal gene programs. So this would be upregulation of various extracellular matrix proteins. There's remodeling that occurs within the heart. So I just showed one image of that, where you start to see disarray within the tissue. There's upregulation with regards to extracellular matrix protein, which is fibrosis. And then ultimately you will start to see some type of cardiac dysfunction, whether it's arrhythmias or changes in fractional shortening or cardiac output. And so what we questioned were, how well could we mimic some of these features within a microfluidic or an organ-on-chip platform? So the basis of the chip is a muscular thin film chip, but we also have additional chambers to this. But for the purpose of this talk, I'll just focus on the cardiac side of this. And so what we wanted to do was really use some of the features that we've seen within the literature, and then challenge our system. And so one of the things that we wanted to make sure that we were able to recapitulate were the actual structural features of the cardiac tissue or the heart tissue. So here is what a native heart tissue look like. So if you take a slice of a heart, this is what you would expect to see. This is our engineered cardiac tissue. And so we want to essentially be able to mimic that alignment that's seen within healthy hearts. And then from that, we want to then be able to stimulate our tissues with various factors, and then assess how well we're able to generate disease-like features. And so to do that, we're looking at gene expression, we're looking at cell shape changes, we're looking at calcium changes, and then we're also looking at contractile function. And so just these videos here, you can appreciate there's differences, even if you're not able to quantify them, there's differences between the healthy and diseased tissue. And so for this study, we decided to use Angiotensin II, and we chose Angiotensin II for a number of reasons. So one being that Angiotensin II acts upon just about any cell type within the cardiovascular system. There is an effect of angiotensin II on those cells. And so this schematic shows that within cardiac tissue, what's been reported is there's induction of hypertrophy and also transdifferentiation of fibroblasts into myofibroblasts. It can play a role in inflammation and ultimately in cardiac failure. But the important thing with angiotensin II is that it's actually important in cardiovascular homeostasis. Even though it plays a role when it becomes dysregulated, it also contributes to the pathophysiological features of cardiovascular diseases. So within our study, we, as I stated earlier, we built these or designed these engineered cardiac tissues. And so one of the ways that we do that is by using a technique called microcontact printing. And so this is essentially what microcontact printing is. So if you've ever used a one of those rubber fake tattoos that you ink the design and then stamp it onto your arm, or if you have a kind of a signature where you have it in a stamp and then you ink that stamp and then place it onto a piece of paper and therefore transferring the ink and your signature onto the paper. That's very similar to how micro contact printing works, in that we have a polymer stamp with a specific design. We coat ECM onto that polymer stamp, remove the excess, and then we invert it onto our polymer coated platform. And based on that, we're able to transfer those designs, those extracellular matrix patterns, directly onto our chip platform and seed cells. As the cells attach and start to spread, they then create these anisotropic cardiac tissues that look similar to what we would expect to see in an in vivo setting. And so one of the first things that we did was quantify or analyze gene expression. And so one of the traditional or one of the characteristics of disease is the reactivation of the fetal gene profile. And so we looked at a number of genes that have been identified within cardiovascular disease. And so what we did were look at over a period of time, up to 72 hours, and we saw the greatest gene changes at 72 hours, and specifically genes related to extracellular matrix protein and also cardiac conduction. So connective tissue growth factor, TBOX5 and SCN5A. So we saw that there was a decrease in SCN5A and TBOX5. So these are genes that are really important in conduction within the heart. And so this was actually promising, and seeing that we were able to mimic some of we were able to induce a hypertrophic gene expression profile within our disease model. The next thing that we did was to look at morphological changes within our system. And so to do this, we again treat it with angiotensin II. So we treat it with two different concentrations, 5 nanomolar and 100 nanomolar. And then we used MHJ to trace these outlines of these cells with individual cells within our engineered cardiac tissue. And we measured aspect ratio, sarcomere organization, and also tissue thickness using confocal microscopy. And what we found was, after quantifying all of these metrics, were that we only saw marginal changes within the tissues. And I'll talk about why I believe that is the case. But once we decided to look at functional, we looked at calcium handling. So within the literature, there have been a number of studies that show that angiotensin II can lead to arrhythmic behaviors in rat models. And so what we did was we treated our cells again with angiotensin II, and then we use FlowFluor as the calcium dye, and we looked at the signal profiles. And so within the controls, we saw what we expected to see, nice normal peaks. But within our angiotensin II treated cells, what we started to capture were arrhythmic events and early after depolarization events. So from this study, we can say that angiotensin II treatment leads to upregulation in these early after depolarization arrhythmic events within our engineered cardiac tissues. And our final assay for this study was actually looking at functional readouts, again, with our engineered cardiac tissues. So within rat hearts, it's been shown that angiotensin II exposure leads to decreased shortening and fractional shortening within rat hearts and also mouse hearts. And so we can't really measure contractility within our system, but we can measure contractile stress generation. And so within our system, what we did, again, using those two angiotensin II concentrations, we were tracking essentially the curvature of these films, so how far the films are deflected. So what causes the films to deflect are we have these PDMS or polydimethyl siloxane films that are coated with an extracellular matrix protein, and then we seed the cardiomyocytes on top of those films. And so as those cardiomyocytes are contracting, that's what's really causing the films to deflect. And so we're tracking the deflection of these films, and then we're able to quantify the stress generation because we know the thickness of the films, and then we can use a modified Stoney's equation to actually calculate the force. And so what we saw within this, and you can appreciate definitely from the movie, so B is the control, and C and D are the angiotensin II treated tissues, that we see a significant decrease in the contractile stress that's being generated by those tissues that have been treated with angiotensin II. And so what we wanted to do then was just really kind of compare our model to in vitro studies and in vivo studies. And so within our system, we're able to see that we saw an upregulation in hypertrophic related genes. We saw an upregulation in early after depolarization and arrhythmic events. And we also saw a decrease in contractile stress, which matched what was seen within in vivo models. But we were not able to capture the morphological changes that you would expect to see during cardiac dysfunction or cardiac failure. And so part of that is we believe is because of the technique that we use. So we're using microcontact printing. So essentially, we're dictating to the cells how they should, how are they going to align and organize within our system. So we believe that's a limitation. And so we're addressing that by looking at if we stimulate earlier than what we did, what we start to see differences in organization or in morphological remodeling within the system. So some of our next steps are that we focus mostly on cardiomyocytes, but we can appreciate that the heart has multiple cell types. And so we're starting to build in different cells within our system. We also can appreciate that these cells can actually sense mechanical properties of their cultural substrate. So now we're looking at mechanically tunable materials to improve upon our model. And we've started some initial studies now with our chip building in the endothelial side of this. And so we look at endothelial response to angiotensin II, and we're specifically interested in gap junction formation. And so for that, we used the coherent. And what we saw was that there were decreases in gap junction responses after a week of angiotensin II treatment. And so we're using the same concentrations that we use within the cardiomyocytes. And so this is ongoing work. So we're just starting now to kind of put the pieces together and start to really build a model that more closely resembles what we would see within portions of the heart. And just kind of to highlight some other things that we're doing within the lab is we're really interested in building in silico models as well to use to inform some of our design decisions. So the idea would be if we can build these sophisticated computational models, then it could potentially help us to kind of guide our decision-making processes with regards to the experiments that we choose to move forward with. And so this was some work that we've published with regards to modeling blood flow within the vasculature, mimicking individuals with sickle cell disease. And what you see here are the blue dots that you see are cells that are adhering to the walls of the vessels. And so we were measuring or mimicking vasal occlusions that are common within sickle cell disease. And we were able to quantify that by measuring the percentage of adhered cells. And what we saw were that really in these bifurcation areas is where you see the highest amounts of cell adhesion. And so that means that that's likely where your highest amounts or numbers of vasal occlusions would occur. So these are areas that are more prone to vasal occlusions. And so this is just some of the work that we've been doing within our team. I thank you all, and I thank my team, and I can take any questions if there are any.
Thank you very much. It was an interesting talk. We have one question from Charles Shang. Does COVID or SARS-CoV-2 cause acute heart failure through angiotensin II receptor?
So I'm not sure about the link between COVID and angiotensin II, but I mean, I have been definitely looking at the studies that have been coming out with regards to COVID and the consequences on the heart. And so that's definitely an area that I'm interested in starting to look into, because I think really considering the consequences of chronic diseases will be really an interesting area with regards to heart disease and heart failure.
Right, and there's another question from Peach Tao. Since neonatal CMs have a greater regenerative ability than the adult ones, if you change the neonatal CMs in this study to adult ones, do you think you'll get the same results?
No, and the reason for that is, for the most part, one of the reasons that we chose the neonates is because we're really dictating to our system how to form these tissues. With adult heart cells or adult cardiomyocytes, you don't have that ability, because that kind of square 7-to-1 ratio that our cells, our healthy cardiomyocytes form, or they're really in that structure. And so it's really difficult to pattern adult heart cells and then to culture them long term. So I would imagine that a lot of the results would be more so the cells having aversion to the system that we have here. But we're actually collaborating with the group at THI to look at introducing induced pluripotent stem cells into the system. So that could be something that would then be something that we could compare against the neonate results that we have.
Okay, thank you. I think we need to move on. I had wondered if you were collaborating with the people at Methodist. We've got 2 heart talks already, so that's great to hear. Okay, our next speaker is Dr. Laura Smith Callahan, and she obtained her BS degree in bioengineering at the University of Toledo, and her MS and PhD in biomedical engineering with a focus in biotechnology and tissue engineering, respectively, from the University of Michigan. During this time, she received an NSF graduate fellowship and an NIH predoctoral tissue engineering and regeneration training position. Following a 1-year postdoctoral regenerative sciences training position, she moved to a postdoctoral training position in the Department of Polymer Science at the University of Akron. Her work at both University of Michigan and Akron has focused on the effects of emulating the size scale, surface chemistry, and modulus of the extracellular matrix. She is now at the University of Texas Health Science Center at Houston, where she holds appointments in the Department of Neurosurgery, Center for Stem Cell and Regenerative Medicine, and the Department of Nanomedicine and Biomedical Engineering. And so she will be talking to us about improving nervous system regeneration using deliverable peptide modified matrices. Dr. Callahan.
Thank you for that great introduction. So today we're going to talk about, if the slides will advance, essentially neuro applications for, there we go, tissue engineering and stem cells. So through the lens of SCI, what happens is you have a mechanical onslaught, whether it be from a fall, a motor accident, sports trauma, or other violence. This sets off a cascade of release of local factors, which eventually leads to the secondary mechanisms that people often talk about. Ultimately, this leads to increased cell death, but even those cells that survive have increased limitations on what they can do due to lack of myelination and other things that really inhibit their ability to function properly as they would in a normal system. Now, currently, there aren't a lot of great regenerative treatment options for this type of injury. So to deal with the axon pathway death, the edema, the inflammation, people have started to use cell replacement to release neurotrophic tractors to change the environment and to help the cells that are spared and remaining function a little bit better. But currently this has drawbacks. So you inject the cells where they're needed, but they tend to migrate very far from where you are. And if they're not in the right location, are they really doing everything they could to bridge the axon, the defect that we have going on? The next thing is once the IPS are injected, they don't really mature, as you see down here, in terms of getting those mature neural markers, and they aren't actually wrapped in terms of the myelin. So these sort of things limit the ability of actual signal transduction through these new established pathways to get us increased signaling. The other thing is that there's lack of interaction between these transplants and the host cells. So it really limits the throughput potential of what's going on. Now, recent evidence does indicate that these cells in this limited form are contributing to the improvements we're seeing. But if we can resolve some of these issues, we have the potential to actually really increase the effectiveness and the efficacy of restoring some of these physiological drawbacks to this type of injury. So the way you kind of think about this, right, is in the body, it's a very elegant system. We have stem cells. They renew to maintain within the system. Then we have, when we need them before injury, the differentiation of the cells, where we need them, when we need them. So we maintain this frequency and potency. Now, we've gotten a lot better over the last decade, but when we get involved in this, we kind of have a mishmash. We don't have the cells, the pure populations that we need, and we don't have limited sort of fates. So we get a mix. We need to get better and more like the body. The other thing is, since the spinal cord is an ordered tissue, we need to look at how does this actually develop. So in the native body, you have pioneer neurons that actually strike out following extracellular matrix cues and then move forward with chemokine stimulates to actually establish the first level pathways. Secondary cells then follow this through ECM and cell adhesion sort of portals to really build out. So how do we emulate this? Well, our approach is going to be a little bit different than a lot of the field takes, which is just to emulate the chemokines. We're actually going to go in, use some of the growth factors, but then string out the peptides in the extracellular matrix so that we can actually have emulation of these extracellular signals going forward to get the cells to follow paths we want and reestablish connections. So why is emulating this extracellular matrix very important? We've had a couple speakers kind of talk on it already today, but if you think about it, the ECM controls everything the cell views. It's their parental control. It's the support structure. It presents the ligands for their attachment. It brings cytokines. Essentially, it filters everything the cell sees and just controls its universe and perspective. And this is different for the tissues, which is how you get different cells to do different things. So from an engineering perspective, how do we manipulate these aspects to be able to mimic the tissue of development that we want to emulate to restore function? And essentially, it boils down to three things we can change. The first one is the mechanics, which is typically referred to as the Young's modulus. Next, we're talking about signaling, and this is ligand density. So ECM, growth factor concentrations, spacing, essentially where they are and how they're presented. The last thing is architecture. So this gets into things of blending and composition of the extracellular matrix. How big are the features, and are those features aligned in the extracellular matrix? So really quickly, we're just gonna tick through each one of these things to show how they're important and what kind of effect that they can have on your cells moving forward. So to do this in a systematic way, we're gonna use a combinatorial method. And fundamentally, regardless of which method you're using, it boils down into one of two scenarios. The first one is an array. So this, you have little spots of different compositions. This sort of system is great if you know if I have A plus B, and I just need to know how much C, and whatever gets me the most C is what I want to use. The next system is a gradient system. This works a little bit better if what you're looking for is a little bit more nebulous, or you're not actually sure what aspect of your cell response is going to be most important, or you're actually looking for multiple responses, because you can look slowly over the change to see how the cells change. So with IPS, most of the time, we're in this gradient scenario because we don't quite know what we're looking for or where sort of the outputs are going to be. So to look at mechanics first, so our Young's modulus, we use this gradient system that you see down here. We can change the composition of these, is visualized through this colorimetric change, but ultimately what we're looking for is changing this mechanics. So we're going from about 500 pascals to about 3,500 pascals. And just for reference, this is between the range from brain up until about cartilage. So the thing to think about most neuroscience and most cultures in general is that they're happening on tissue culture plastic or glass, and that's really in the range of bone. And from the work of Dennis Disher, about a decade, actually about 15 years ago almost now, we know that these changes, these gross changes in mechanics, can have huge effects on the lineage choice of our cells and how they behave. But what we did is we came in and looked at a much smaller regime, which people didn't think cells were quite as sensitive as that. So what you can see is over a 500 kilopascal change, we have gross cytoskeletal changes in terms of the structure of the cells. And this correlates with mRNA expression for cytoskeletal markers, TUJ and MAP2. So these small changes can really have significant outputs on how cells behave, how they align, and sort of the neural network outputs you lose. Moving forward to ligand concentrations, we're going to look at IKVAV, which is essentially a signaling element for laminin that's thought to replace most of its biological features. We advance the system from being cells on top to being cells encapsulated. And what you can see is down the length of this material, we can change the IKV concentration along with a degradable cross-linker from collagen that degrades in response to MMP expression. Typically, what's thought to happen with this is that as you increase the degradable cross-linker concentration, you should see increases in axon extension. But since we're also increasing the IKVAV, what we discovered is in terms of axon extension for the human IPS cells, is that IKVAV signaling dominates this. So even though there's greater degradability in the matrix, we're not seeing that axon extension. So that gets us to our optimal concentration of IKVAV in our system, which is very similar to what we previously found in mouse models. So we know that IKVAV isn't enough. So we're actually adding a second element that was originally isolated from laminin, but is in a number of other biological proteins, and that's LRE. So you can see that it has a similar concentration dependent effect on cell attachment and axon extension. But when you add it with IKVAV and block its signal, you can see it has significant effects on morphology and then also the attachment of the cells. So we're using this combination going forward, but we need a platform to put it on. So in this set of studies, we developed a three-dimensional hyaluronic acid matrix. And all we're changing is the amount of reactive elements. Both of these scaffolds have the same amount of hyaluronic acid in them, but all we're changing is the number of reactive moieties. Just doing that has significant changes for mechanics, but also the axon extension of the cells and the mRNA gene expression of maturation markers over time. So just for the rest of this talk moving forward, this is going to be the language we're using. DIF-HA is just the bare reactive moieties on the hyaluronic acid. PEP-HA is our hyaluronic acid with the IKVAV and the LRE signaling tethered to it through covalent bonds. Now to make a 3D structure, we're reacting both of these with methacrylated hyaluronic acid. So the first thing that happens is when we just coat this on a surface, like many of the things we've looked at today, and seed mouse neuronal stem cells on them. See, we have long organization areas. So if you look at, just for reference, the scale bar on the top is 1 millimeter. So these are long areas of organization. And you can see from the bottom breakouts that they're much more organized on PEP and do not exist at all on just a bare laminin surface, which is traditionally used for 2D culture. When we measure the axons, we find that on the PEP surface, axon length in the alignment is significantly longer than either the DIF-HA or the unorganized laminin. This also corresponds to an increase in cytoskeletal expression of neuronal marker TUJ. So we're organizing the cells, we're affecting the cytoskeleton just by coating the PEP-HA with these two peptides in an optimized concentration on the surface. What gets really interesting is we take that same base level cell, but we just change the chemicals that we're putting in the media. So instead of driving it to be a neuron, we're driving it to be an astrocyte. And what you see is that alignment doesn't happen in these cells when they're in the different chemical media and differentiation process. But so that is a good indicator that it's not topography, it's not actually surface roughness that's driving that organization, it's chemical. The next thing we see is drastic changes in the expression of astrocytic reactive markers. So we see a decrease in C3, S100B, and viamentin, and the viamentin is even reduced during standard collagen insertion. This has significant implications for essentially astrogenesis. So moving forward, we wanted to look at this plasticity that we're seeing in terms of a more specific marker for it in the astrocytes, and why all the cells are expressing GAP43. We see higher fluorescent expression collaborated with higher mRNA expression on the cells in the PEP than we do on the diff or the laminin. So this has further implications for astrogliosis, microglia activity, and neurological toxicity as we move forward into our animal models. So what we did here is we did a contusive model of spinal cord injury. After 2 weeks, we injected our matrix. In this particular set of pictures on the left, the red is our matrix 4 weeks after implantate or injection, and the green is neurofilament. So what you can see first is we have a lot more HA that seems to be hanging around from our matrix in the PEP relative to the diff. But we have good interaction with what's remaining of both matrices and the neurofilaments. Overall, when you look at the whole total spinal cord, this leads to a trend in increase in neurofilament. So we have more neurons theoretically left, and we have a slight increase in plasticity. So in PEP-HA, we can see that this is partially double stained with the neurons. So it's an indication that they're actually extending, but there's another cell type there. So looking at this injury a little bit more closely, what we see is increase in GAPGH, so our astrocyte marker, paired with nestin, which is a neural stem cell marker, but at times immature astrocytes actually will extend both markers at once, which is something that we didn't have time to talk about, but we actually observed in the 2D studies we were looking at a minute ago. What's important here is even though we have increased astrocyte markers, we don't have increased extracellular matrix. And when you look at a close-up of the extracellular matrix, you can see DIFF-HA tends to be far more packed and congealed together, so it's far more dense, where we have greater spacing and less density in the areas where we have PEP-HA. So it's an indication that going forward, we might have less maturity in terms of what we have going on. So because of that changes in extracellular matrix, we wanted to look at metastatic proteases. So these are things that actually degrade matrix. MMP2 is something that's associated with regenerization and axon extension. So what you can see is that overall the expression patterns seem to be different in our injury model. We have very unspecific, untargeted administration in the areas in the DIFF-HA, but the PEP-HA, it seems to be following lines, which is similar to what you would see if it was promoting axon extension and activities like that. So we wanted to back out and see what was going on in the 2D models with this MMP2.
2 minutes.
So we have high expression in the astrocytes, but this correlates at this time point with decreased mRNA expression. So it's likely winding down on the PEP. At day 5 here, we don't have expression on the neurons, but if you look at earlier times where the network is not fully formed, we do have high expression, which makes sense because this is actually correlated with network formation and not network maintenance. Looking at it in 3D to go deeper in the neurons, blocking, which is this bottom panel here, MMP activity, actually limits axon extension. So MMP is playing a vital role in that network formation, but not a significant role in the maintenance. We also find that the PEP-HA, this optimized signaling, actually plays different activities in activation and total presentation by the cells, indicating that there's some level of regulation and control not observed with the DIFF-HA. So with that, just like to review what we've talked about in terms of mechanisms, signaling, and computation of your matrix has significant influence on your tissue response or your cell response, and can actually be used directly to manipulate cell behavior. In our optimized system of IKVAV and LRE, we found differences in alignment extension and expression of neural and astrocytic markers, along with astrocyte activity, which then led us to find differences in tissue response in a subacusis spinal cord injury model. So with that, I'd like to thank the members of my lab, my collaborators, and my funding sources, and I will be more than happy to take any questions.
Thank you very much. It highlights, this is a pretty complicated process.
Yeah, it is.
I'm impressed with the responses to the peptide HA. Were you anticipating those? All the changes you've seen?
We anticipated some of them. The actual effects on matrix degradation were something that it took a lot more piecing together of the literature to sort of identify. And it actually relates back to a lot of what we talked about today is LRE, one of its targets is a calcium channel. And that directly manipulates a lot of the MMP signaling.
Okay. Okay, so at this point, there are no other questions. There's a little lag. So there may be questions that will appear in the next minute or so that perhaps you can go to Brella to answer. But thank you very much for another nice presentation.
Thank you for the opportunity.
Our next speaker is Jeff Jaycott. There he is. I see Jeff. Just by introduction, Jeff is currently at the University of Colorado in Denver. He received his education and training originally, I guess, undergrad in Colorado at Boulder. From there, he moved on to BU in Boston, and then to UCSD in San Diego. And he's a bioengineer, I guess you could say, and those are really tremendous places for bioengineering. So he's really a well-trained individual, that's for sure. Jeff was here in Houston for a number of years at Rice, and has then moved back to Colorado. He's received a number of awards, a career award from the NSF, as well as other awards I won't go through. So yeah, I think I'll just let Jeff take it from here. Jeff, welcome back, and look forward to your talk.
Great. Am I on? Okay. And it looks like my presentation is on.
Yep.
So thank you very much, Jim, for that introduction. And it's great seeing people on here again who I remember from Houston. Yeah, I was at Rice for, I think, 9 years, which was great, and then decided to move back to Colorado. And right now I'm in the Department of Bioengineering, which was started, I guess now 12 years ago. I had a task force and wanted to have a department and brought people to the
You're fading in and out.
Over an old it's the in the state, although there are degrees.
Jeff, you're fading in and out. If you want to turn off your video, I can share your slides for you.
Am I fading in and out?
Yes.
So the video is fading out, so just my slides a little bit.
I can't hear you. Cannot hear you. Oh, that's better. Turn off your video and then Dawn can show your slides.
All right, if I stop my video, can people hear? I still can't hear me?
Not really.
I think that's a little better. If you want, I will share my screen. I have your slides pull up. Just say next slide when you're ready for me to go to the next one. Is that okay?
Yeah, that could work.
Okay, give me 1 sec.
Is this any better?
Not really.
Can people hear me now?
A little better now. Yep.
Okay, I have your slides up if you're ready. Just say next slide.
Okay. Well, Jeff, we can't hear you at all. Maybe
I'm not sure if anybody can hear me now.
I can hear you now.
But okay, Jim can hear me. All right, great. Yeah, so let's go to the next slide. I'll try to go through this quickly and just let me know if my voice still fades out. The motivation behind this is that about 10,000 infants in the United States are born with a congenital heart defect, and many of these need to be repaired soon after birth. Generally now they're repaired with either polymers and artificial materials or things like fixed pericardium. So originally, tissue material that's then been fixed with glutaraldehyde and really contains no live cells in it. So in the next slide, I showed that these can often just become scar tissue and leave areas in the heart that are not contractile and non-functional and really greatly increase, especially the chance of arrhythmia, but also other heart function, and can greatly increase the chance of sudden cardiac death. So just to go with a big overview of kind of the vision that we have in my lab, if you can go to the next slide, I think, right? I can go through it here, see what is next. Go to the next slide, and you can just go through all the stuff that's in there. But we've looked at and we focused a lot on cells that we can get from a fetus that's diagnosed with a congenital heart defect that can be used in the infant for repair of these structural heart defects. And the vision is that about 80% of congenital heart defects are diagnosed prenatally in a fetus, and we're hoping that we can isolate stem cells, differentiate and seed them, and then use them for surgical reconstruction. So we've collected these. You can go to the next slide, and we show it the karyotypically stable. And I like the slide because it's a male fetus, so it has a Y chromosome, so we know we didn't get maternal cells. And I have something that maybe is later on that. And then the next slide, we analyzed these. They're mostly mesenchymal. We can differentiate. Well, they're mostly mesenchymal. They don't have hematopoietic markers to them. You could skip through all of those. We've been able to get them from amniocentesis, and then I think the next slide, I'm not sure what I've uploaded here. So, and I think I'm kind of off on slides. What we've shown is that we can also get these from doing manual rupture of membranes for induction of labor through vaginal birth, and can get these at the time of birth too, and have done this analysis here that shows, here you could see 6 markers from maternal saliva, and the fetal saliva contains exactly 3 out of the 6, so 50%, which is what we'd sort of expect, and we don't see any maternal contamination even collecting it there. So we could show IPS reprogramming, which as you hopefully could see here, I might be out of date. Let's see. Am I still coming through okay? Can I still share my slides? Or maybe not. Am I still breaking up?
No, you're okay right now.
Okay. Let me try this.
I stopped sharing, so you can try.
Okay. Sorry about that. I have no idea why it suddenly started breaking up. Okay, cool. So here is showing that we can get, I guess I passed over the slide, the IPS induction slide, but we can get very high, in fact, higher IPS induction of these than in almost any of the other cell types that the labs that we're collaborating with has looked at. We could go through cardiac differentiation, and I have a lot of markers here, and get high differentiation of these. Let's see. And I always love things like this because I can open our incubator and watch them all beat. So I quickly put this in, which is why I wanted to get to these slides, just based on some things from talks this morning. And this is some, older work that I've done, but I think it kind of relates to a lot of what people are looking at with matrix effects. We look at matrix, but another thing that we've really focused on is the mechanics and mechanical stiffness. I know that Dr. Smith-Callahan was talking about this earlier, effects on cardiomyocyte maturation. And you can ask questions about this later, but I'll go through this real quick. But I've found really that there's these key regions where the cardiomyocytes will start to mature. And if it's really loading on them, if the myocyte's unloaded, or if it's isometric contraction and it can't move, they won't really mature, especially not even developing a sarcoplasmic reticulum, and calcium handling would be greatly changed in these. And we could show that this is all signaled through Row-Rock pathways. But what I really wanted to get into here is a lot of what we've looked at in trying to make these scaffolds in laboratory grown heart tissue that we feel that we can implant and then can incorporate with native tissue. So the first couple slides are showing our initial studies looking really in vitro at how we can best make something using both natural materials and using supporting layers that can have the right mechanics for a slide, but for a patch, for something that can completely replace a ventricular wall, or be in place of myocardial tissue, and what really has effects on these. So this is one that I wanted to comment on, because we'd seen that other talk this morning on heart matrix, where we've shown that if we add heart matrix to these, and these are hydrogel patches that are attached to and supported by a thin polymer membrane that gives it the mechanical support, that if we add heart matrix, we get stronger contractions, and we get faster conduction velocity, although still slower than in native heart tissue. And then another thing that we've done, and this is all in vivo studies, is looked at how we can even increase the cell-cell communication and the conduction velocity and have used carbon nanotubes and found that we can put those in at a level that's not toxic to the cells, but that greatly increases by about 3 times the conduction velocity, getting to conduction that's seen in native heart tissue. So now going to the in vivo experiments we've done, and most of this was done by Peach Tao, who's here in the audience and has been asking questions and could probably answer questions on this also. But doing this patch, this drawing that I have done is a little incorrect. The patch is sewn into the incision line, and so it actually becomes part of the heart wall. So we could do this surgery where we replace about the top half of the right ventricle freewall with something that we've made. And so far, we've put in things that aren't contractile, but really just looking at what the ideal makeup is of a material we put in to get any sort of muscular and vascular ingrowth. And have shown that if we don't put any cells in and do this surgery, that we still get better, well, we get cells invading into this. And what we've used here is a fibrin-based hydrogel supported by a polyurethane layer. And we get improved heart function when we put this in, and especially can greatly reduce arrhythmias compared to fixed pericardium, and then can also show much less fibrosis with a fibrin patch compared to fixed pericardium. And some regenerative remodeling, some ingrowth of cells, but not all that much. So we wanted to look at how we could improve on that. And oh yeah, this shows RV function is better with these hydrogel patches than with fixed pericardium. So then we really started looking, and this is spanning a bunch of years between Rice and Colorado, at differentiating amniotic fluid cells into cells that could be vasculature. So we have shown that we can differentiate just by using very high levels of VEGF. These stem cells we get from amniotic fluid into what looks like arterial endothelial cell. And by taking these fibrin gels, that we cross-link them with polyethylene glycol to reduce their degradation. And when we do that, we can put in, we figured out we could put in endothelial cell and then these unfractionated amniotic fluid cells as support cells, and really get a capillary system that forms through this. And if we implant this into a nude mouse, we get very fast vascularization and attachments to the mouse vasculature. So then we went on to looking at this in a pre-vascularized patch. So we take these PEG fibrin gels supported by polyurethane because we need something that really gives it strength, and again use it as a replacement for right top of the right ventricle free wall in this rat model. And when we do that, we get greatly improved, significantly improved right ventricle function compared to the same thing without a pre-vascularized system. And really thinking that the vasculature is what's needed to pull in the other cells. So we could show here that a lot of this seems to mediate having a vasculature in there seems to mediate the immune response. If we look at leukocytes and pan macrophages that invade, and we do get vasculature not nearly as close to what you would see in a native heart tissue, but still some vasculature throughout, and muscle cells that do invade through into these areas in this adult rat model. So with, let's see, So that's, yeah, all I have to go through. I was going through it pretty quick, so ask questions, but I think I used up a lot of time just trying to figure out the technical aspects here. But looking at stem cells, we can isolate from amniotic fluid that we think can be used for repair of structural congenital heart defects shortly after birth. Looking at these vascular patches that we can make, and then really showing what happens when we put in this really kind of immature, rudimentary, but still pre-vascularized capillary-like vasculature in these, and then implant it, taking place in the heart wall. And so here is, my lab a couple of years ago, and all of the people in it. And, we have a lot of funding sources that we got back from Texas Children's and Rice, and now lots of others. And we're working with Children's Hospital Colorado and the Gates Center for Regenerative Medicine here at the medical school campus at University of Colorado. And with that, I'll take questions. I see I have one here asked. And then anything else that comes up too.
Thanks, Jeff. Is there a question? I don't see it. Is it on chat?
There's a question in chat from,
Yes.
yeah, George Eisenhofer. Oh, no,
Okay.
maybe that was an answer. Oh, that's an answer in chat. I just
Okay.
it just appeared.
Anybody have any questions they'd like to ask Jeff? So since you're kind of the expert in, or an expert in delivery here, I mean, what do you think is the best way to deliver things to the heart? Is it patch?
Yeah.
Patch is the absolute best, or what are your thoughts on that?
Well, as far as delivery, there's different things that you're looking at treating. Patch, so when you're looking to change heart structure, or expand right ventricular outflow tract, or patch septal defects, then a patch or some sort of three-dimensional tissue, I think, is the only way you can really do that. And that's what we've been focused on. Cardiomyocytes have, in our group and most other groups, been very difficult to keep alive, no matter how they're delivered to a heart. And that's one reason why, these studies that have really worked, and these results here, have focused on the vasculature, which can be delivered and can stay functional, whereas the cardiomyocytes, that's still really a challenge, especially when you have something that's thick and that's only attached at the edges, that you want to keep viability of these functional cardiomyocytes. And I think that's a big challenge going forward. But otherwise, there are methods. If you look, some recent things that have come out of Chuck Murray's lab at University of Washington, of injecting myocytes, IPS derived and embryonic stem cell derived myocytes into hearts, have really, that's really all I've seen that has gotten really good survival of those cells. Whereas, most methods to do that, the cells die really quickly.
Okay, thank you. Are there other questions? If I could throw one more at you, I guess since you're focused on pediatric congenital heart disease, and you're looking at single ventricle patients, these hypoplasts. I mean, how do you think I'm just trying to understand, how do you think your patch approaches would work in such a severe condition like that?
Yeah, so a lot of that's what has really driven this, that we think, starting with something like we're starting with a patch, and then going from there to something that can form a whole ventricle. And I think that would be the ideal result of this. I think that starting smaller is necessary just to get that to work before trying to do a whole ventricle. But we're looking a lot at that. We're looking at Yeah, many of the cells, in fact, that I've shown you that were used in these in vitro studies came from patients with HLHS, so kind of a single right ventricle disease, that we're really trying to see also how do those cells function. We're using these as model systems, which I haven't shown any of that data, to try to figure out how mechanotransduction happens and how heart structure functions using specific patient cells that have some specific um, heart condition, structural heart defect.
Okay. Well, that's terrific. Well, I guess if there are no more questions, I don't see anything else. So thanks very much, Jeff.
Thank you, Jim.
And we'll move on to our next speaker, Rahul Roy.
Hi there.
You're here? Great.
Yep,
Okay,
yep.
so just by way of introduction, Rahul Roy is a Rice bioengineering alum and a Texas native, and he's an expert in 3D printing. He's currently at CellLink, and I think he's going to be telling us about some very sophisticated bioprinting and how it's connected to regeneration. So welcome, and please take it away. Thank you.
Thank you very much. When I tell people I'm involved in 3D bioprinting, I'm frequently asked if I can print a new heart, lungs, or liver. And while for now, the answer is not quite, recent advancements in bioprinting have end-use relevance for patients today, and they bring us one step closer to creating complex whole organs for regenerative medicine. Founded in 2016, CellLink has evolved from a bioprinting-focused company to a bioconvergence company, meaning it offers tissue engineering solutions up and down the workflow. And as a result, it has expanded with team members and customers across the globe. Now, as for where CellLink fits in the current medical landscape, pharmaceuticals take on average to develop to the tune of $2.6 billion. And while there are many factors that contribute to this cost and timeline, bioprinting and in turn CellLink promise to reduce inaccuracy and in turn improve efficiency. Breakthroughs that allow bioprinting to play a greater role in drug development will fundamentally allow it to take on the longer-term challenges of organ shortages. Now, looking at the pharmaceutical workflow can show how 3D bioprinting will come into play. The process typically begins with high throughput screening assays at the molecular and 2D cellular level. And here, bioprinted model assays will better filter compounds that will allow progress to the next stage. At the preclinical level, the ethics, expense, and at times inaccuracy of animal models demand that they only be used as much as absolutely necessary. Comparisons to bioprinted models can highlight sources of error stemming from physiological differences, lab environments, and simply procedures. And then finally, at the stage of human clinical trials, markedly better upstream data will result in better outcomes for participants and better preparation for adverse outcomes. As in vitro platforms increase in complexity, more and more of the traditional biopharmaceutical workflow can be replaced. Now, focusing on the issues with 2D cell culture, studies have shown that both healthy and diseased cells behave differently on their own and when exposed to drugs in 2D versus 3D conditions. Not to mention, the typically stiff substrates on which 2D cell culture is performed typically influence gene expression, morphology, and viability. And considering how strongly cells are influenced by their environment, it stands to reason that surrounding cells with a more familiar 3D cellularized environment will produce more physiologically relevant results. So the success of bioprinting depends on the interplay between the fields of cell biology, material science, and mechatronics engineering that serve as the basis for 3D printing. As 3D models become more and more complex, increasing the functionalization of materials, and as well as the cell biology that informs these selections, will become more and more critical. However, today's applications particularly focus on how architecture influences the success of the printed construct. So it's most important to understand how these technologies work and therefore bring these structures into being. So numerous 3D bioprinting technologies have sprung up over the last 30 years, but CellLink offers three, each with its own advantages in terms of resolution and flexibility in terms of the number of materials that can be used and the variety of said materials. Starting with the BioX, it's an extrusion printer, which applies air pressure to a cartridge full of gel, liquid, or melted plastic that is pushed through the nozzle, seen in blue, into a strand. And then stacking these strands layer by layer will result in your printed construct. The simplicity of this approach makes it compatible with a wide variety of biomaterials and solidification types. And it's starting over. There we go. The Luminex is a DLP-based bioprinter developed in partnership with Houston's own Volumetric Biotechnologies. A light source, in this case a visible light projector, shines a sequence of images like a movie into a droplet of light-sensitive liquid that, wherever the light touches, solidifies. Stacking these cured images results in the printed part. And although only one material can be used at a time, it's a high-resolution approach that can be used to create clear, watertight microfluidic devices or lattice-like structures like the one shown here that are absolutely impossible with extrusion. And here you can see it in action. So the Luminex uses single photon polymerization. Since the light source is the correct wavelength and energy to solidify the material right where it makes contact. With that process, the curing is strongest when the light reaches the surface, and then the power dissipates further on. The Holograph X, on the other hand, developed with Prellis Biologics, uses a laser light source at roughly double the wavelength needed to solidify the material. So it passes through the focal point without interaction. But then a simultaneous pulse of light causes a jump in energy at the focal point, such there is sufficient energy to cure the material. And you can see this on the right-hand side image as the little orange dot next to the 2-photon absorption arrow. With this technique, capillary scale curing can occur anywhere within the volume of the photocurable liquid as opposed to just at the surface. Then to take it a step further, a device called a spatial light modulator can multiply that single point of curing into tens of thousands of points of light at the same time to rapidly increase the rate of printing. Compared to traditional 2-photon printing approaches, we can get prints up to 88 times faster than just the single point alone. And so with these technologies in mind, we can go back to the comparison of 2D and 3D cell culture. Some in-house studies that were done with the BioX, GELMA, and collagen-based bioinks were used to qualitatively analyze cell behavior in 2D versus 3D. So starting with some HUVEX, you can see some nice sprouting after 14 days of culture. Stem cells after 14 days managed to preserve pluripotency as evidenced by these particular markers. And then lung cancer cells in 3 dimensions appropriately localize this particular marker on the periphery of the cells instead of just completely throughout in 2D. And then you have liver cells that are correctly expressing actin at the borders of the cell clusters instead of, again, just kind of everywhere. And so if we take this modeling a step further, we can now compare the response between the 2D and 3D drug conditions. But before that, on the right-hand side, you'll notice that the timeline for 3D models is substantially longer than that of 2D. However, automation and parallelization offer avenues to dramatically reduce the impact of those first 6 days on the drug development timeline. And then, presumably, the potential for increased accuracy resulting in fewer failed trials should make up for the rest of the time. And here you can see what is involved in that 3D model workflow, just like before, with the lion's share of the preparation time occurring between the BioX printing and the IDOT drug dispensing steps. And so specifically, the work was done with false positive and false negative approaches or scenarios. In the false positive scenario, which in real world practice would lead to a dead end, potentially costly, avoidable animal study, cisplatin dramatically fails to inhibit pancreatic cancer cells within the 3D model, suggesting it should not be pursued further. However, the false negative scenario represents a lost opportunity for potential therapeutic good, as gefitinib here demonstrably impacts cancer cell viability in 3D. Now, using 2PP technology, the prior studies were performed with avascularized 100 microns spheroids. But if we're going to look for a more accurate organ model, larger vascularized spheroids would paint a more predictive picture of the actual physiological response. And so using a basket-like structure printed with the holograph X creates these tubular channels that are gas and small molecule permeable that can serve as vasculature for these spheroids that are instead 500 microns, even up to a millimeter in diameter. And Prellis once again did a false positive scenario using cisplatin, except this time with breast cancer cells. And then the false negative scenario in this case analyzed prostaglandin's effects on cells exhibiting a KRAS G12 allele. While the prostaglandin demonstrated some efficacy in the 2D culture, a more pronounced effect could be seen in the 3D culture, which would in turn influence the dosages used in subsequent trials. Now, turning our attention towards customer and other partner-driven applications, we have a Rensselaer project that seeks to create artificial skin grafts using a collagen-based ink. And although they were aiming for in vivo applications, conceivably grafts made using this technique or similar techniques can also be used in vitro toxicology studies. In fact, the FDA is using a BioX to examine this very thing. And so based on the results of their efforts, you can see the development of the dermis and epidermis. Oops, the slides went a little bit out of order. Vascularization over in this side, and the epidermis and dermis on this side. Relative to the other work that I have shown and will be showing later on, this publication is a little bit older, 2018. However, it demonstrates really nicely how a very simple model can be used to show a critical physiological phenomenon. So this heart valve model from Harvard is responsible for trying to reproduce the mechanical gradient seen in heart valves, explicitly aiming to model a fibrotic condition down the road. Not only were they able to reproduce this gradient, but they also successfully modeled a fibrotic response. Super simple model, but very good for preliminary evaluation of drugs. Now, this particular work is interesting, not because the BioX directly produced the biomaterial or printed the biomaterial, but instead they used the BioX to extrude sacrificial channels that were then covered with a microfluidic device that they built. In this case, they were using tumor spheroids next to some sort of vessel to investigate how the tumor cells responded to cells that were inside that vessel. And more specifically, they were analyzing how ovarian cancer cells would migrate in response to a process called neutrophil netosis, including analyzing how neutrophil proximity, quantity, and presence of flow affected this migratory response. Not only did they demonstrate a correlation in netosis to the cell migration, but they also showed that the type of cell migration was in the therapeutically more challenging clustered form instead of just through single cells alone. So again, that can better inform in vitro different kinds of drug targets, or just a better understanding of how this metastasis process works. Now, it would be remiss of me to go through this entire presentation without highlighting the incredible work from Miller Lab, especially the alveolar construct that demonstrated in vitro oxygenation. Besides this work leading to the development of the Luminex and the line of photoinks, it also opens the door to intricate hydrogel microfluidic devices where, unlike with PDMS devices, the bulk material can actually act like a physiologically relevant participant of the study, just like you can see on the right-hand side with the hepatic animal study where they built a GELMA construct and embedded it. Embedding the cells inside the bulk of the material simply would not be possible with a likewise PDMS device. I'm really excited to see, based on this work, what kinds of platforms people develop. And one such platform comes from Emory and Georgia Tech. Using image data from a variety of modalities, embryonic and fetal heart models were created and computationally analyzed to see how fluid flow affected the development and viability of cells. As Dr. Jacott mentioned, the challenges with dealing with cardiomyocytes, for this preliminary study, they stuck to using HUVEX just to generally see how it works. But in the future, they're hoping to improve the model with more relevant cardiac cell types. And so they basically lined the walls with the HUVEX, and they showed that the HUVEX took quite well to their models, expressing appropriate markers, morphology, and then proliferating. And of course, another step in the future would be to thin out the walls of this bulk material and better match the actual morphology of the developing heart. And so in terms of future directions in general, of course, improvements are going to continually be made in terms of the quality and complexity of these models. But in addition, there are improvements to be made around the rate of production and validation of these models. As I mentioned earlier, the timeline for preparing a 3D model is significantly higher than that of 2D. So Celllink's acquisition of an automation manufacturer like Genolis allows for mass manufacture of ready-to-use bioprinted models, which would save on time and cost. And so effectively, that upfront 6-day timeline wouldn't really need to be factored in to the drug development lifecycle. And then with the acquisition of Mattek, introducing 3D printing technology can increase the complexity and accuracy of the models that they already manufacture. And they have a 25-year experience with industrial needs so that they can ensure that any 3D printed models that are created will have the level of consistency and accuracy needed to meet cGMP requirements. And then in the far future, maybe we'll be able to have full-size organs. And so in summary, while full-size organs are still on the horizon, 3D bioprinting can still benefit patients today. Bioprinted 3D cell culture models will improve results up and down the pharmaceutical workflow, and clearer developmental direction will offset additional model and prep time. Parallelization and automation are also great avenues to offset this particular prep time. If you want any additional information, also you can visit cellink.com, where we have a number of application notes that talk about the in-house work that we've done or work that we've done in collaboration with our customers. And finally, I would like to thank our customers, colleagues, and partners, without whom none of the work that I presented here today would be possible. And with that, I'll open the floor for questions.
Terrific. Thank you very much. I guess there's one question. Please, I'll read it to you. Please give suggestions for CellLink bioprinter users on how to increase cell viability.
Okay.
Yeah, go ahead, take it.
So I guess it depends on the bioprinting technology. Of course, if we're talking about extrusion, which is arguably the most common one, shear is certainly a known issue for extrusion printers. And so allowing yourself to use a larger nozzle diameter, as large as possible for your particular application, can certainly help. Having a lower viscosity can limit the shear on the cells. And then, I mean, after the printing process, it really strongly depends on particular applications. So I suppose if someone is having particular issues with cell viability, they can definitely reach out and we can figure out what would work for their specific needs.
Great. I don't see any more questions in the chat. Are there any other questions? No, I guess not. Well, okay. If there are no more questions, then thank you very much.
Thank you very much.
Wonderful talk. And I think I'll turn it over to the lunch.
So now we'll move to the next talk, which is going to be given by Henry Justino, a board-certified pediatrician and pediatric cardiologist. So we've gone from what's on top of the heart to the heart now. He's a professor of pediatrics at Baylor College of Medicine and directs the cardiology innovation and co-directs the Mullins cardiac cath labs at Texas Children's. He's going to talk to us today about development of a transcatheter polymeric heart valve, and clearly going to give us the scoop from bedside to bench and back again, because it's never a straightforward path. Thank you.
Henry, you're muted.
Okay, thank you. Can everyone hear me now?
Yes.
Wonderful. Thank you so much for the introduction. And what a wonderful talk, Louise. Thank you so much. So I'll talk to you today a little bit about the path we've taken to develop a transcatheter polymeric heart valve and how to take that from the bench, from the bedside to the bench. And now we're on our way back again to the bedside, hopefully with the device ready for clinical trials very soon. So let me begin by showing you my disclosure slide. The only one relevant to this talk is the bottom one, that I'm a co-founder of a company called Polyvascular that I'll be talking with you about, that now is the one developing this transcatheter heart valve. So a little bit of background first for those who may be less familiar with congenital heart disease. It is the most common of all birth defects we encounter, and it's the #1 cause of infant mortality in the developed world. In fact, twice as many kids die from congenital heart disease than all childhood cancers combined. So it's not to say one disease is, more important than another, but it is a substantial cause of morbidity and mortality in the developed world. Now, congenital heart defects come in many different types. Some are the types that are considered simple defects that can be cured with a single procedure. For example, a hole in the heart that we can close with open heart surgery with a patch, or what I do for a living as an interventional cardiologist is to place a device in one of those holes and close them with the device. And that's a cure. Generally speaking, kids don't need another procedure after that. But other heart defects require multiple procedures and often multiple open heart surgical procedures over their lifetimes. And that's where we decided to focus our energies to tackle one of these very vexing problems in congenital heart disease, which is the problem of poor durability of replacement heart valves. So to just take you through a little bit of the normal heart, as you know, there are 4 heart valves, and we're going to be focusing predominantly our attention on the pulmonary valve. So the pulmonary valve is a little bit of a forgotten valve when it comes to general cardiology, because it's uncommon for adults to require work on their pulmonary valve. As we all age, our mitral valves and aortic valves require attention, and that's an acquired form of heart disease. But when you are born with a heart defect, you are much more likely to be born with a pulmonary valve defect compared to all other heart valve defects. So On the far right, what we see is a heart that that has developed abnormally. The pulmonary valve, instead of being, as on the left, a nice tri-leaflet valve with a wide opening to send blood to the lungs, instead we have a valve that is completely blocked, or at best a pinhole in some cases. And there's also a hole in the heart here, a ventricular septal defect. So we call this condition pulmonary atresia because the valve is completely occluded, or atretic, as we say. And a child born with this type of heart defect will undergo open heart surgery to correct the defect, which includes closure of that hole in the heart and placement of this valve here in light blue, as you see. This would be the first of many replacement heart valves that the child would need throughout their lives. Now, one might think that we have lots of options for what kind of valves to give to children. It's not that uncommon a problem after all. And yet there are really very, very few types of valves that we can give to a baby to place in this position between the right ventricle and the pulmonary arteries. And by far and away, the most commonly used valve around the world, and including in the US, is a human cadaveric valve, what we call a homograft valve. That means a valve derived from another child that died and donated their valve, so we can use that spare part in open heart surgery to give to another child. It seems almost unthinkable that we are still doing this, because this hasn't really changed for decades, but that's what's the status quo, and really very little has changed since then. So what is the problem with giving someone a cadaveric human valve? It should be wonderful. It's a human tissue valve, so going into another human should be the ideal replacement. In fact, it's not. It's the worst thing we can do. The valves have very, very poor durability, and within a few months, they shrivel down and become severely narrowed or severely leaky, or both. Again, a valve, because it opens and closes in flaps like this, really only has two functions. It's got to open well and it's got to close well. So when a valve is dysfunctional, we say that it's dysfunctional by having developed narrowing or failure to open well, or by leaking, which is failure to close well, or by both. And these valves frequently develop both, but narrowing is a big problem for them especially. The additional problem with giving homografts to children is that these human cadaveric valves are actually in short supply. If you imagine that you have a 2 or 3 month old baby that's undergoing this operation, there has to be a child of similar size having donated their valves. But fortunately, there are not many infants dying around the world to donate their valves. As a result, on our shelves here in our hospital and everywhere in the world, these human cadaveric valves are in very, very short supply. So we may have a valve that maybe the ideal size for this baby might be a 9 or 10 millimeter valve, but we might have an 8 or a 14 because of what was available in the children who've died in this region and who've donated their valves. So it really is problematic, and we are often having to place valves that are missized for a given child because of their limited availability. But the bigger problem I will focus on is the first, which is the poor durability of the valve with the valve shriveling up like this. When valves deteriorate and have to be replaced again, that means many children all around the world are having to undergo yet another open heart surgery to keep replacing these valves that have quite poor durability. But taken collectively, that paints a very significant and sort of grim picture of what these kids have to go through. But if you look at any individual child, that's really what they're going through. They're going through a lifetime of these valve replacements. It's not just a one-time thing. They'll have their first valve when they're a baby, and then another valve when they're a toddler, and then another valve when maybe they're in grade school, another valve when they're teenagers, and then this doesn't stop when they're teenagers, because the valves don't last forever, that we place them even if they're adult-sized. They still deteriorate over time and have to be replaced over and over again, on average every 10 to 15 years. So that's a lot of open heart surgeries for these patients. Now, why does all this happen? It turns out that human tissue is actually a very poor substitute to give to another human, because of the fact that we express HLA antigens on our cells, and these lead to an immune response in the recipient of those homograft valves. And these antibodies are formed in our bloodstream and attack the valve and destroy it and cause it to become severely calcified and shrunken down. And these antibodies can persist for up to 15 years after an operation. So we develop lifelong, if you will, immunity and memory of having been exposed to human tissue. So this is a study that was done looking at the incidence of the presence of these panel reactive antibodies. So we can test a person's blood for the presence of these panel reactive antibodies against a wide host of HLA antibodies from the general population. And before an operation, most people would have very little in the way of circulating antibodies against other human tissues out there. But within a month, we have very, very robust antibody response. And within 3 months, basically everybody who has received one of these allograft or homograft, meaning human cadaveric valves, has developed 100% panel and reactive antibodies. Just to give you an idea, that's after one operation. But once you receive a second or a third valve, because there is crossover between the HLA antigens being expressed in one human to another human, a second valve is likely to express some of those same antigens and yet other new ones as well. So now the antibody response will be heightened, and the rejection of the valve can be sometimes worse the second time around, and the third time even worse. So this is a graph showing what happens when you receive one of these valves and how long it takes before the valve is sufficiently dysfunctional that we say, let's go ahead and replace the valve, okay? So at about 6 years, only about 80% of patients who have received one of these valves still have their valve. It's been removed in about 20%. But that doesn't tell the whole story. Freedom from reoperation, as these curves are often termed, because these Kaplan-Meier curves denote how long it takes before how many people are free from an operation at a given number of years post the first surgery. It doesn't tell the whole story, but this tells the story, which is the freedom from valve dysfunction. So it turns out that more than half the valves become severely dysfunctional by 1 year. We don't replace them at 1 year, we replace them much, much later, which means we're basically living with a severely dysfunctional valve for all of these years, because it's so unbearable to have to put someone through yet another open heart surgery 1 year after they receive the valve, that we just say, well, just live with it. And we try to palliate that in the cath lab with ballooning open that valve and things like that. But really, eventually, those valves do need to be replaced. So the median time to failure of the valve is really 6 months if you look at regurgitation. And if you look at failure by means of stenosis or narrowing of the valve, failure to open, it's about 13 months. But the median time to reoperation is 40 months. So again, we limp along with valves these kids limp along with valves that are really poorly functional. Now, the immune response to these homografts is something that leads to about 60 to 100% of all of these homografts being replaced within 5 years in anyone who's received one of these valves in their first year of life. And another untold part of the story, or largely untold, because there certainly are papers describing this, but we have not yet really fully grasped the importance of these studies. Dr. Shaddy has done a lot of this work and tells us that really what happens when you develop these reactive antibodies to human tissue is that you can become ineligible for receiving other human tissues. So far, I've made the point of that being another homograft valve. But in reality, there are much more important things that we have to give people. That's entire organs, kidney transplants, liver transplants, heart transplants, etc. And so we have children, we have one in our hospital right now, a teenager, who solely because he received 2 or 3 operations to receive human cadaveric valves, because that's the standard of care around the world, he is now ineligible for heart transplant. And his heart has failed, and he's been living for years attached to a machine, a ventricular assist device, and that's been his existence because we have not done our job of coming up with better substitutes than human cadaveric valves to give children with congenital heart disease. So clearly, repeat operations to keep replacing these valves over and over again is a poor idea. It puts the patients at greater risk and discomfort. It's a longer recovery time for these families to have to see their children go through. And the valves have poor durability, and the whole process leads to immune sensitization. So is there a better way? Well, there's already a better way, and that's the transcatheter pulmonary valve replacement. So already there are animal tissue-based valves that, instead of being delivered by open heart surgery, are delivered by a minimally invasive procedure that is catheter-based. But there are still significant drawbacks. The valves are really suitable only for teenagers or adults. No one is making a valve available for small children, and they're quite expensive and difficult to manufacture from these animal tissues, which leads to other problems. So let's talk a little bit about transcatheter valve replacement. Many are familiar with the concept of what we call now TAVR, transcatheter aortic valve replacement. And yes, the aortic valve is being replaced at very rapid rates around the world as this technology has become widely adopted now as a less invasive way of replacing an aortic valve. And although that's now very, very common, that wasn't the first transcatheter valve replacement. The first was actually a pulmonary valve in a child, in a 12-year-old child. And that was done using a bovine jugular vein. So cattle and other animals that graze have well-formed jugular venous valves, so that when they bend down to graze, the blood doesn't rush back into their heads. And one of the physicians, Dr. Bonhoeffer, thought that this would be a good substitute to put in the pulmonary circulation. Venous circulation is a low pressure circulation, and the pulmonary circulation is a relatively low pressure circulation that was thought to potentially do well with this kind of low pressure valve. So he took the jugular vein of a cow and hand sewed it onto a metal frame that we call a stent, and collapsed that onto a balloon, and then delivered it into a child's heart to act as a substitute valve within a degenerated valve the child had already received. So the old valve served as a landing zone to deliver the new valve into it. And this worked really well. And over several more years, this became the Medtronic Melody valve. It was bought out by Medtronic and is now sold as the Melody valve. So the valve, as you can see here at the bottom, is hand sewn onto a metal frame, and it's put onto a balloon catheter, which is collapsed onto inside a sheath. The sheath is 22 French, so it's about a 7 to 8 millimeter thick catheter that goes into the groin, maybe something about the thickness of a pencil or so. So it's a relatively large bore catheter, and so not really something for very small children, but for older children and teenagers, it's become a real advance for them. So this is an example of a teenager with one of these valves, a Melody valve that's going to be placed. This is the echo showing his valve that has a lot of narrowing and some leak back. The red backflow there is the leak. And he had elevated pressure in the right ventricle because of that narrowing, that kink caused by the shrinkage of that homograph valve. And this is what it looks like when we take a picture in the cardiac cath lab. We're injecting dye in the main pulmonary artery, and the valve is this narrowed area here that has a little bit of leak back. This is a front view, this is a side view. And this is a procedure done by one of my colleagues many years ago as we were learning to use the Melody valve. This is the Melody valve that was collapsed onto that balloon. And this is now that balloon expanding to open up that metal frame. And inside it, there are leaflets of that bovine jugular vein. You can see the valve still has quite a narrowing. The stent does not fully expand. And this is what it looked like afterward. There's no more leak, and this all looked very good. But 7 months later, he came back worse than what the condition he was in when we first treated him. And he developed even higher pressure in the right ventricle because now there's an even worse narrowing located at the valve itself. And in the cath lab, this is an angulated view looking down the barrel at the valve, and it's no longer cylindrical. It's more like oval, it's crushed, it's collapsed itself because the metal frame has fractured. And so this is something that we were able to fortunately treat for him by placing two very, very sturdy stainless steel stents inside that Melody valve to restore the cylindrical shape that it should have. And with that robust metal, place yet another Melody valve inside the first Melody valve. So this is called a valve-in-valve placement. And normally it's not done only within 7 months, but this is what it took for this patient. And now he's been many years out and doing very, very well. And that's what it looks like after the valve placement. So why did this happen? Because the Melody valve is well known to have developed stent fractures. And this is what we learned and everyone else around the country. And these are x-rays showing the metal frame completely fracturing in multiple places. And that's because of how brittle the metal is, but also because of the recoil forces of these homograph valves is tremendous, and they want to shrink back down to that original shriveled down size, so it can crush the whole valve. Another major drawback is endocarditis, which is a heart valve infection. And the Melody valve has unfortunately been implicated in what appears to be a higher than usual incidence of endocarditis compared to other heart valves. And this is a nice normal looking Melody valve, very thin leaflets, delicate leaflets. This is what a valve looks like when it has developed endocarditis. It is completely eaten up by pus and bacteria or filling this whole area. This can be a life-threatening emergency that requires an operation, sometimes even in the middle of the night. So it has led to many centers now believing that the Melody valve should not be used, and the valve is falling rapidly out of favor. So we really have very few options now, and in some senses, there are people saying, should we just go back to doing homograph valve replacements for these patients, which of course is a terrible plight that they have to go through. The other major drawback of any of the current technologies that are out there for transcatheter valve replacement is their manufacturing needs. So this valve requires 15 hours of human labor to trim the fat off it and hand sew it stitch by stitch all along every part of the metal here. And in the process of manufacturing these valves, 95% of the raw materials of the source jugular venous valves are discarded. So it's incredibly wasteful, incredibly expensive. And as a result, there is almost no penetration of all transcatheter heart valves of any type in low to medium income countries. So at best, we are developing with transcatheter heart valves a first world solution for a very limited segment of the population that would need them, because we don't have valves that go to the sizes we need for small children and even bigger adults that don't have them available either. So, and animal tissues are not free of the development of an immune response. It also occurs to animal tissues. So I'll move ahead a little bit to talk about where we have come from and why we went into all of this, is really to develop, to completely rethink heart valve development. We decided that we would develop a valve that would also be delivered by a minimally invasive catheter-based approach rather than open heart surgery, but do so with a valve that is made out of a medical grade polymer rather than any human or animal tissues, that would have very simple manufacturing technique to make it actually affordable around the world, so that people, even in developing countries, could have access to a quality heart valve rather than either having to do open heart surgery with homograph valves, or even failing a proper system of harvesting human tissues and processing them in some developing countries. They just have access to nothing and they don't survive.
Good morning.
Thank you. And we've even developed the valve to be redilatable over time to grow with the child. And our value proposition is, again, the cost saving to the healthcare system. This is our team here with some of our students working with us. We went through the TMC innovation system. This is our valve operating in a pulse duplicator, showing it opening and closing well. It already meets the International Standards Organization requirements for valve performance. And I apologize that this is not playing right, but this is the valve working inside the accelerated wear stations. The valve is already manufacturable in a variety of sizes, ranging from children's sizes to adult sizes. And we have just completed a 3-year chronic animal study. This is the valve being prepared on the delivery sheath. This is a sheep pulmonary artery in our preclinical studies. And this is our valve being expanded in the sheep pulmonary artery. And immediately the valve is functioning well, and we have no leak, and the sheep are up and about within a few hours. Now, we've, as I said, completed a 3-year chronic animal study showing that the valves are performing beautifully with almost no narrowing, almost no leaking, and just explanted our valves. In terms of the market opportunity, there are about 1,000 patients eligible for the Melody valve based on the limited sizes that cows come in their jugular veins. And because of our ability to make valves that are much smaller or much larger, we could probably have a market that's 2.5 times larger to serve many more children and adults that would need these types of transcatheter valve replacements. And this platform technology really lends itself well to the development of other types of heart valves that could be made out of medical grade polymers, as we have done. So these are the founders. I'm Henry Justino. Kwan-Soo Chun is a PhD mechanical engineer. Dan Harrington is a material science engineer. And the three of us founded Polyvascular several years ago. We have assembled a scientific advisory board and many advisors to work with us. And we were fortunate to have quite a bit of traction when we came out. We won the top prize at the American College of Cardiology Innovation Challenge in 2018. And I would have told you a couple of months ago that everything we accomplished so far, including all the benchtop design, testing, and then animal study, was done in about $1 million worth of entirely grant funding. But I'm very pleased to tell you that we now, through a phase 2 SBIR grant, have been able to now expand our team. So Will Clifton has just joined us as our Chief Operating Officer. Many of you may know him through his connection from having been at Procerion and a co-founder of Inventure. And Steve Andrea joined us as well from Adient Medical before that. And this SBIR grant is currently a $2 million phase 2 SBIR grant that will allow us to continue this development and then translate that into the first in human early feasibility study. So I want to thank all of the research supporters that we have here. This all began with an anonymous donor who was very gracious and gave us a little bit of money to get us started and generate some pilot data. And then from then on, it's been entirely grant funded with no dilutive investment funding at all. I also want to thank all the students that work with us throughout the Texas Medical Center and all the and all the mentors that we've learned from who so patiently taught us along the way. So I want to thank you so much for your attention, and I'll be happy to take any questions you have.
It's a pleasure now to introduce Cindy Farrar-Carson, who's going to give closing remarks. And Cindy is a professor at the University of Texas Health Science Center at Houston, and is one of the best people I know at building partnerships in this area. I can't think of a better person to sum up the day.
Great, thank you, Doris. And I agree, this was a wonderful day. We were really disappointed when we missed our original date with the freeze, and we're all still dealing with our frozen trees, but it turned out great. And I just want to take a few minutes and thank some people. So first of all, thanks to all of you who joined us today and listened and asked questions, and I hope you enjoyed it as much as we did. I want to thank our keynote speakers who were absolutely inspiring. I want to thank all of our speakers and our poster presenters. I encourage folks to continue to collaborate and work together and follow up on anything you heard today that's interesting. I want to thank our sponsors. Without them, we can't do what we do. I want to thank the GCC staff, Suzanne and Shannon, and especially Dawn today for heroic efforts in keeping us all functioning on these virtual platforms that are not the easiest things for some of us to use. And just to thank all of you for the help you give us all year round for all the things so that we can work together. And finally, I just want to invite everybody to our next conference, which we don't know when that will be yet, but hopefully it will be in person. That is what I am hoping for. Get your vaccines and hug your friends is hopefully in all of our future. And shortly after, I think we'll be announcing a series of networking events, and we'll keep you on the list for those as we, sort of move toward more normal operations, and maybe we can all raise a glass together at some point soon. So anyway, I just want to thank everybody again and hope you enjoyed the day.